transfers a pair of electrons from NADH to ubiquinone, with several redox centers acting as intermediate acceptors. First, 2 electrons are donated to a flavin mononucleotide group. From here, the electrons are shuttled one at a time through a series of iron-sulfur sites. Finally, ubiquinone accepts the 2 electrons in turn to become fully reduced. Ubiquinone is a mobile electron carrier that is freely diffusible within the lipid bilayer. For every pair of electrons transferred, complex 1 concurrently translocates 4 protons from the matrix to the intermembrane space. A proton wire is formed as the protons are rapidly relayed through hydrogen-bonded amino acids within complex 1. In this manner, a proton differential, and therefore a pH gradient, begins to form across the inner membrane. The energy released from the dissipation of this proton gradient will eventually be used to form ATP. 2 other things contribute electrons to the transport chain. The first is succinate dehydrogenase from the citric acid cycle, sometimes referred to as complex 2 in the transport chain. The second is the glycerol phosphate shuttle. These are not discussed in your textbook as part of the electron transport chain because they do not directly contribute to the proton gradient. Reduced ubiquinone next shuttles its electrons to the mobile carrier cytochrome C through the action of complex 3. Cytochromes are proteins containing a characteristic heme group. Members of the cytochrome family contain a characteristic heme group that consists of a porphyrin ring surrounding a central iron atom. The iron cycles between oxidized and reduced states as electrons are passed. Some cytochromes are part of large protein complexes, but cytochrome C is a peripheral membrane protein that acts as a mobile electron carrier. Complex 3 extracts electrons from ubiquinone in a detailed 2-step process known as the Q cycle. 1 electron is passed to an iron-sulfur protein, to cytochrome C1, and then to cytochrome C. The second electron follows a different route, passing through cytochrome B and then back to a quinone to produce a semiquinone. Another ubiquinol donates its 2 electrons to complex 3. 1 electron passes through the iron-sulfur protein and cytochrome C1 to cytochrome C, and the other electron passes through cytochrome B to the waiting semiquinone. The net result is that 2 electrons are passed to cytochrome C and 4 protons are pumped into the intermembrane space to contribute to the proton gradient. The final step in the electron transport chain is the reduction of molecular oxygen by electrons derived from cytochrome C. Cytochrome C ferries its electrons to complex 4, which contains both heme prosthetic groups and copper ions. Ultimately, 4 electrons must be transferred through complex 4 to result in a full reduction of oxygen to water molecules. The net result is 4 more protons are pumped across the inner membrane per molecule of oxygen reduced, or 2 protons per electron pair. We have shown